Okay, welcome to part 2 of this video series on advanced microscopy experiments in fluorophore design. So in our previous video, we talked about GFP and different types of genetically encoded fluorophores and how they function in the actual microscope and how they're able to emit certain wavelengths of light to be able to show us specific protein interactions. So today what we're going to discuss is a bit on how do you actually design a fluorescent system and how do you actually get fluorophores onto protein and targets of interest. All right, so fluorophore engineering. So there's many different techniques available to label your sample. Some of the most common are CRISPR engineering of molecules of interest into living organisms. So again, these are proteins. We know their genetic sequence. And so we can use CRISPR to actually append these onto into the DNA of the actual system that we want to look at. We can do exogenous protein expression. So using a plasmid vector system, we can introduce a engineered protein design to some sort of bacteria and then purify out the protein, which we're going to go into detail on how you do that as the second part of this video. You know, my apologies. One of the easiest ways is actually just using an antibody. So you can conjugate a fluorophore to an antibody, and then that antibody has a specific epitope that it's labeling against on a protein of interest. So you take that fluorescent antibody, you throw it into your system, and then that epitope will bind to your protein of interest or whatever your structure of interest is. It doesn't have to be a protein. And then you can get expression of a fluorescence on a certain molecule. You can also use a primary antibody with no fluorescence and then a secondary body with a fluorescence to bind to that primary antibody. This antibody labeling methodology is highly used in flow cytometry, so not just through a microscope, but flow cytometry is using fluorescence and fluorescent antibodies to tell you what cell types you have. So if you're interested in seeing a CD8 versus CD4 T cell, you can make an antibody specific to the type of receptors that are available on these T cells. And then when you throw them through the flow cytometer, they'll pick up different fluorophore colors for different T cells. And then the third is fluorescent dyes. So these are not proteins, these are chemicals that are meant to interact with and stain different motifs inside of your cell or whatever your interest system is. These dyes are usually not very specific, so that is usually a downside to them. But the upside to them is they're usually really easy. You generally just throw it in there and you're done. Like I said, this is a chemical. This is not an actual protein. Dyes are chemicals that are meant to interact with things. So I use a lipophilic dye that interacts with the phosphodiester backbone of things to label them. And this is a highly sample dependent process. Some things work better than others. If you're using an in vitro system, you're likely not going to CRISPR anything into your in vitro system. You're going to use a conjugated protein that was exogenously expressed, and you're going to throw it into your in vitro system. Antibodies don't penetrate cells very well, so a lot of times you need to use a CRISPR system in a living cell to make it so that you can actually view things. So it's really dependent on what you're trying to look at and what type of system you're working with. All right, so method number one, CRISPR engineered cells. Again, this is very common. I'm sure you've read a paper on this at some point of how CRISPR engineered cells are able to show you things. These are largely used for live sample applications. So if you're looking at mitotic systems and you want to see things in cell division, you might CRISPR it in a fluorophore to a protein of interest that you're able to see when it's in mitosis. The general broad strokes approach: identify your gene of interest, and then you insert your fluorescent protein to either the C-terminal end or the N-terminal end of the protein of interest, depending on what part of the proteins are functional in your system or not. And then you just tack on a fluorophore at the end of that sequence, and you're going to get a conjugated, genetically encoded fluorophore that's expressed in the cell. And so then, when the gene is expressed, the protein from that gene will now be fluorescent and we can see it on a microscope. For example, this is one of the most powerful ways to look at things like genetic expression vectors. Are things active all the time? Are they silenced? Are they upregulated, downregulated? One of the best ways you can do that is by just looking at the cell and seeing when things fluoresce. The second method is an exogenous protein expression. So this is really only good for in vitro assays. You're generally not going to throw an exogenous protein into a living cell and have it be happy. But when you're looking at an in vitro system, it's really powerful and really easy to use. So the approach is you're going to design a plasmid with your favorite protein, and then you're going to insert a fluorophore to either the C-terminal or N-terminal end, just like in CRISPR, and then you're going to express the protein in some sort of vector. Most often is bacteria, because bacteria are cheap. E. coli is well understood, and E. coli does a great job of taking up plasmids and making things. The downside of bacterial expression vectors is a lot of times it doesn't actually work. One of the limitations you have of that is the folding machinery that you have available in that system. So if you have something like an E. coli and you're trying to fold a really complex protein, a lot of times you're going to end up with a misfolded protein that is insoluble and just makes aggregates, which is what you don't want. But it's really dependent on the protein that you want. Another common way that it's done is you use the embryos of your model system. So, for example, Xenopus laevis, I can CRISPR in a protein into Xenopus laevis embryos, and then those embryos can be ground up and I can purify out the protein that way. That's usually a lower yield and more expensive way to do it, but it usually works really well if you need specific protein folding. And then once you've got your favorite protein expressed, you need to purify it. And so we do this through purification columns. Histidine purification is a very, very common way of doing this. You get beads that have an affinity for a 6-histidine tag that's added onto the C or N terminal of your protein, and then those histidines will bind to the bead. You run it through the column, and then you wash it out, which I'll talk about a little bit more later. So method number 3 is antibody labeling. So this is mostly used in like a fixed cell environment. You're going to need to permeabilize your cell so that you can actually get your antibody into it. You're usually going to need to do some sort of blocking assay where you stop any activity happening. The good thing about antibody labeling is that it's usually very specific. If you have a good antibody, you're able to see things very specifically inside of your cell. The downside is what you're looking at is generally dead. Right? It's a fixed cell assay. And so your cell is dead. It's no longer functioning. You can't see changes. But you can get very high resolution and very discrete labeling of proteins of interest by using antibody affinity labeling. You can also do what's called dirty antibody labeling, where you throw in an antibody to a living system, either in vitro or in vivo. The downside of that is you're not going to have any sort of antibody block. So you're going to get off target, and you're most likely going to get off target effects of your antibody. But usually, if your antibody is good enough, you'll get more on the thing that you care about than the background off target thing that you don't care about. And so you can make that distinction between them. General approach is going to be you're going to grow an antibody against your protein of interest. This makes antibodies very expensive. Antibodies are by far the most expensive labeling technique you can do, and that's because we need a living organism to be able to express an antibody. Most commonly, antibodies are grown. The bigger the animal, the less specific it's going to be in general. So if you're looking at a monoclonal antibody that's just got one target, they're going to grow it in something like a donkey, right, which is going to make a lot of antibody, but it's probably not going to be as specific. So a lot of times what they'll do is they'll grow a primary antibody in a donkey, and then they'll grow a secondary antibody in something like a rabbit, which is smaller, but it's going to give you more specificity. And they're going to grow that secondary antibody to be specific to the primary antibody that was grown in the donkey. So you're going to get more specific antibody labeling by using this dual antibody approach. And it's expensive, and it's really only good for fixed cell environments. You can get away with it in vivo, but it's going to be much more effective in a fixed cell environment. And then when we add our secondary antibody, that's where we're going to add our fluorescent tag in the dual antibody approach. So fourth method, chemical dyes. So again, these are usually like lipophilic dyes, or they're not specific to a protein. They're specific to a chemical signature of proteins in your model organism. So again, lipids, even like actin motifs, things like that. It's going to be very broad strokes that are going to dye things generally, but not specifically. These are generally just industrial design small molecules with fluorophore attached. So the purification approach for this one is you're going to pull out your credit card and you're going to purchase it, right? This is not something you're going to synthesize in your own lab to be able to be used. And then dyes are designed to interact with specific chemistry inside the cell. So they're chemistry motifs, but not specific proteins. I hope that's clear on the differences between like dyes and the protein levels. Is one is against a chemistry environment, one is against a protein environment. And like I said, I've highlighted down here. Dyes are not proteins. They are chemicals and small molecules that interact with chemical profiles in the cell. But one of the really nice things about dyes that can make them very powerful when used in conjunction with more specific labeling techniques is if you have like a lipophilic dye, and I use that as my example a lot because I use one. It's called DIO. A lot of times they're cell permeable, right? They're small molecules. So if you want to look at like lipid structures inside of a cell, you don't have to come up with a fluorophore that's going to be localized to every lipid in the cell. It's a lot of lipid, right? And there's a lot of off-target effects you can get by trying to design to something as diverse as lipids. So you can add this lipophilic dye to your cell culture medium, grow your cells in it. That dye is going to permeate your cell and pass through the cell membrane without having to do a passivation or electroporation or things like that to actually get it in there. It will just passively happen. And then you can dye your large structures like your lipids inside the cell and then have a secondary fluorescent molecule like a protein or an antibody that looks at a specific structure. So don't rule out dyes just because they're not specific. They have their place. They're just going to be a little more niche, I would say, than the actual protein-specific labeling methodologies. Okay, so now that we know how to make fluorophores, let's talk about an assay that we can do with one. So FRAP is a very common technique that is used in microscopy. It stands for fluorescent recovery after photobleaching. This is what this primarily looks at is protein dynamics. So looking at things like lipid rafts and how they move throughout lipids, looking at like proteins that are localized to a specific area through transport or diffusion, one of the two. But this is a localization assay is really what it gets down to. And so what you're going to do is, again, in this example, we have this nice green GFP cell, and we're interested in looking at this structure here. So we're going to hit this thing with a 405 nanometer laser, which is the way our microscope representative explained it to me. 405 is a nuke, and it's just going to kill off any fluorophores inside this region of interest. So you're going to draw a region of interest in the software. You're going to tell it hit this thing and make all the and burn out all the fluorophores so that they're no longer functional. And then you're going to blast this thing. And then post-bleach, you're going to end up with a non-fluorescent area because all the fluorophores have been burnt out of it. And then we can start a time lapse, and we can look at the degree of recovery of that fluorophore into that location. And so if you have a highly mobile protein of interest or lipid or anything like that, what you'll see is that as a function of time, you'll actually get more molecules to come in and replace those bleached out molecules that you had. So this is really useful in localization assays. Let's say, for example, you have a protein that is known to interact with the centrosome of a cell. So you can go in there with your labeled protein, you see the centrosome, you know where it is, you blast it with light, and then you start a time lapse, and then you watch the rate of recovery as dynein or a different transport molecule brings it to that centrosome, you can then calculate the rate of recovery, which then tells you something about the rate of action of whatever the protein is you're looking at. The next one is called FRET. This one is a more difficult assay than using just FRAP, but it's also a very powerful assay to look at co-localization of two proteins. I'm going to hopefully not lose you in the details of this. FRET is not a super easy concept, but I think we can make it work. So what you're going to do is you're going to get two proteins of interest. So we're going to say we have protein A that we think interacts with protein B. We're going to come up with a labeled version of protein A and a labeled version of protein B. They're going to have different fluorophores on them. That's important. They are not going to have the same excitation profile between the two fluorophores. And you'll see why. So we have put on A, we're going to say we put a 405 fluorophore on there. And on protein B, we put a 477 fluorophore on there. And so we're going to hit our first protein with our 405 wavelength. And when we do that, all the proteins are getting exposed to 405. But because protein B excites at 477, we're not going to get this protein B to be fluorescent. We're only going to get 405 to be fluorescent. And so in the event that you have co-localization, you're going to utilize the Stokes shift and the emission spectra change from 405 to actually excite the acceptor of which is our 477 nanometer in Venus, right? So you hit protein 1 with 405 light. It undergoes a Stokes shift to, I'm sorry, I've been saying this wrong. 528 is our protein B. Protein B is labeled as a 528 nanometer molecule. So our emission from 405 undergoes a Stokes shift, and we don't quite get up to 528, which is fine because these things have what's called off-peak excitation. So our off-peak excitation overlaps with the emission spectra of 405, so we're emitting at 477. Well, the second protein can be excited by 477 even if it's off-peak, so you're using the emission of your original protein to activate and excite protein B. So just going through it one more time for a little bit more clarity. So we hit our proteins of interest with 405. Protein A, which is specific to 405, is going to be excited by that 405 wavelength. It will undergo a Stokes shift and emit a photon at 477 nanometers. Our protein B is going to be excited by those emitted photons from protein A. Protein B will be excited by the emitted protons from protein A. So then when the protein B accepts those photons from A, it will now be fluorescently active. And we can use this property to look at co-localization. So what this is showing here is that these two proteins are too far away from each other for the donor emission to make the acceptor emission fluoresce. So we have no interaction between these two proteins. They're not co-localized. But in this scenario, our proteins are close enough together, they're localized to the same spot, that the overlap of the donor emission can excite protein B's fluorescent molecule. So we see fluorescence in this channel. And this is showing you as well, right? These are too far apart. This thing doesn't become active because it's too far away from this one. And then here, where we have FRET, we have activation of 405. The emitted activates our protein B, and we get fluorescence. Hopefully that makes sense. FRET's a little weird, but really what it comes down to is you're using the emission spectra of one protein to excite another protein. And then you can see the fluorescence of that second protein, which tells you they're co-localized to the same spot. Okay, and so now I want to go into a little bit of a deeper dive on how do we design a fluorescent protein. Right? And so I happen to come across a protein that I'm interested in expressing and labeling with a fluorescent molecule. And so in my design of this protein, I decided to just bring you along in the journey with me. So the first thing we did was I identified a protein of interest. So the CDR2 is a dynein adapter protein, which is allowing different cargoes to bind to a subunit called dynactin, which then interacts with dynein, which allows it to be transported to the minus end of a microtubule, which you guys probably don't care about, but I'm still going to explain it because I think it's cool. And so I'm going to use this probe in Xenopus laevis extract. So we're using this in an in vitro system. So the best way for me to do that is to make a purified protein construct that I can then add into my extract system. So I identified the gene of interest in Xenopus. So it's a highly conserved sequence, but I still want to limit the amount of cross-speciation variation you can have in genes. So I found the Xenopus sequence of the protein. So I have the amino acid sequence here that I'm going to use to generate my plasmid, which I'll then express in yeast, and then I will have an endogenous protein that I can add into my cytoplasmic mixture. So the next step was I needed to identify a fluorescent protein. In my system, I use a lot of different colors. I have a far red protein, I have a blue protein, I have a green protein, I have a regular red protein. So I'm using this M scarlet fluorophore here that's going to be going to excite a different emission spectra than the other proteins labeling that I already have in my system. And so I was able to go to FP base and find my amino acid sequence of my protein of interest that I can then express inside my plasmid and conjugate it to my protein of interest. So then I just went to SnapGene, and what I'm showing you in maroon, or I guess that's more magenta, I'm showing you magenta, is the sequence of my protein, and then this blue region is a linker protein region of the protein, which is just a bunch of glycines with a serine that allows flexibility in the hinge of the protein to be able to be conjugated to my specific protein of interest. And then I have my red protein here. On my protein of interest, I have a functional end to activate dynein, which is the N-terminal end. So that's the end that I need to make sure is functional and not disturbed by appending a gigantic bulky R group onto it. So I'm adding this protein to the C-terminal end of my construct so that my N-terminal domain is not interfered with and can still interact with dynactin and activate dynein. And then once you've done that, you need to find yourself an expression vector that is going to do what you want it to do. So I'm using a PET28C plus expression vector. These are well-designed, well-characterized, commercially available vectors that people use all over the place for lots of different purifications. These vectors are specific to the organism that you want to express in. So the PET expression system is used for E. coli. But note why I decided to use this specific vector is because it has a couple things I want. One, it works in E. coli, which is important to me. Two, it's designed to add things onto the C-terminal end of the protein when it's expressed. So it's going to actually be using the C-terminal end, which is the end I don't care about, and my N-terminal end is going to be conserved. And that's all based on the cut sites and where it's adding things. And then this right here is the business end that I cares on the C-terminal end. It's going to add this 6X histidine tag to the C-terminal end of my protein. So my tag is actually going to be on my fluorophore itself that I can then use to purify this protein out. There are other PET expression vectors that will do this on the N-terminal end, and it's relative to where they put the 6-histidine cut site near your protein of interest. One will have it be designed so the 6X's are added onto your C-terminal end, and then you cut after the 6-histidine, and then the other ones will have it on the N-terminal end, and you'll cut after the 6-histidine on the terminal end. So this histidine tag relative to your cut sites of your plasmid is what matters. So the final step will be to purify my protein of interest. So the way I'm going to do this is I'm going to take that plasmid that we just designed. I'm going to express it in E. coli. There's usually some sort of antibiotic resistance encoded into the plasmid that you care about, which then allows you to treat your colony with antibiotics so that you only get the E. coli that have expressed your protein of interest left after you've done growing them up. You will then take these things and throw them into a test tube, and you'll lyse the cells, you'll break them apart, and then that will determine your solubility, and you run out your proteins on a gel to make sure that they're there. You can look at the specific molecular weight of your protein. So we know that my protein will be the reported molecular weight of the gene plus 5 glycines in a serine plus my gene of interest. So I can locate, using an SDS page technique, I can locate the presence of my protein. This step really isn't technically necessary, but it does save you time and resources. But we know this thing works, and so now we're going to throw it into a main bulk culture. We're going to grow as many of these E. coli expressing my protein of interest that I can. And I'm just going to let these things grow until they're mature. Then I'll isolate them, break them apart, and then once I've lysed them and I've got the intercellular components, I'll run them through a purification column, which is this fractionation collector. And this is where you have beads inside the purification column. And these ones they use, I believe it's a nickel metal that's conjugated onto the bead that is able to have affinity and bind to, covalently or ionically bond to the histidine residues on the end of my protein, and so that my protein will stick to my beads inside my purification column when I run my cell lysate through my purification column. I can then wash the beads in a specific buffer that will cause the beads to release the proteins that was on them, and then I collect them using a collector. And I can use UV detection of my molecule to determine if my protein of interest is in that collection. And then once you've done that, you run another gel to make sure your protein of interest is actually there. And then I have a purified protein that I can add to an in vitro system that I can then image using the microscope. So, once again, I hope you enjoyed and you learned something about protein design and purification, and I will see you in the next one.